A single degree of warming can cut global wheat yields by roughly 6%. During flowering, extreme heat can destroy nearly half the harvest. But some wheat varieties survive these temperatures far better than others. Now, a new study published in Scientific Reports reveals why. Heat-resistant wheat appears to reorganize entire cellular stress networks to stay alive under extreme temperatures. When wheat experiences heat stress, its cells face two major threats at the same time: protein misfolding and the buildup of reactive oxygen species such as hydrogen peroxide, key indicators of oxidative stress. Plants do have defense systems against this damage. But scientists still don't fully understand how these systems coordinate under real field conditions. To investigate this, researchers compared two wheat genotypes: MISA 2, a heat-tolerant cultivar, and Line 4, a heat-sensitive line. The researchers simulated heat stress by delaying sowing by 57 days, exposing plants to significantly higher temperatures during flowering and grain filling. Both wheat lines experienced oxidative stress, but their responses quickly diverged. MISA 2 maintained substantially higher grain yield under heat stress, about 390 grams per square meter compared to roughly 273 in Line 4. The tolerant wheat also accumulated higher levels of soluble sugars and proline, supporting osmotic adjustment, a process that helps cells retain water balance and stabilize proteins under heat stress. One of the most important findings involved structures called peroxisomes. Peroxisomes help detoxify reactive oxygen species and maintain cellular redox balance during stress. Under heat stress, MISA 2 maintained more than twice the peroxisome abundance of the susceptible genotype. The researchers then identified three major regulatory hub genes: TAHSB70, TOSI81, and TOPEX11.4. In MISA 2, TAHSB70 increased about 4.5-fold, while TOPEX11.4 increased roughly 5.5-fold, specifically in the heat-tolerant genotype. Importantly, the susceptible line 4 showed no significant change in TOPEX11.4 expression. Together, these genes formed a coordinated protective module linking protein stability, antioxidant defense, and organelle dynamics. But the biggest difference was not a single gene. It was the entire network. The heat-tolerant genotype formed a highly organized and polarized regulatory architecture. In contrast, the susceptible wheat showed a fragmented network with weakened coordination between stress response pathways. Some genes even reversed their behavior between genotypes. Genes like TASORD and TADERP5B acted as what researchers call phenotypic switch nodes. Genes whose relationships with yield and stress traits changed completely depending on genetic background. In other words, susceptibility was not simply a weaker response. It was a breakdown in coordination between antioxidant defenses, protein quality control, and cellular stress signaling. This study provides one of the clearest systems-level views yet of how wheat cells coordinate heat tolerance under field conditions. Instead of relying on a single heat resistance gene, thermotolerance emerges from dynamic communication between multiple stress response networks. The findings also identify potential molecular markers for breeding climate-resilient wheat cultivars, an increasingly urgent challenge as global temperatures continue to rise. From cellular networks to global climate systems, this is Environment Eyepiece.